Thank you for the introduction. And, I'm a molecular biologist, and I work at the Nano Innovation Lab at Elettra Sincrotrone in Trieste. I would like to thank you for the opportunity to be here to talk about my results. Today, I will talk about two atomic force microscopy applications that involves the use of artificial lipid bilayers for studying interaction of proteins, molecules, or structures like extracellular vesicle with this kind of systems. We are mainly an atomic force microscopy laboratory for biological application. So the main problem in this kind of interaction is represented by the tremendous complexity of plasma membranes in terms of lipids and protein content, and also the dynamics of this structure. So together with the limitation of techniques that are available to study in living systems this kind of interactions, for those reasons, several kinds of artificial systems have been developed in order to overcome these kind of problems and to simplify this kind of investigation. The model membranes is a great tool in the field of artificial systems because you can use these kind of systems to mimic and simulate the cell membrane compositions. You can control very well composition, size, and geometry of these kind of systems, and you can also assess the role of the single components in these kind of membranes. We have different kinds of possibility working with the model membranes. We have vesicles, liposomes, floating monolayer, and supporting lipid membranes. In our case, we used supporting lipid membranes for studying the lipid phase separations and also for studying protein-lipid interactions. These kind of systems can be coupled very well with atomic force microscopy that allows to visualize and measure the topography of model membranes in liquid environment at the nanoscale. And in this framework, very interesting topics is represented by lipid rafts, which are functional microdomains of plasma membranes characterized by a specific lipid compositions that in fact are enriched in cholesterol and sphingolipids, and they are involved in a lot of biological processes, acting as a singularly platform concentrating proteins in the plane of the bilayer. But they are also involved in disease like cancer and neurodegenerative disorder. In this context, I would like to talk about two possible AFM and model membrane application. In the first part, I will talk about the interaction of alpha-synuclein, as the Dr. Ruggeri talked before in his presentation. And the interaction of this protein, that is the main protein of Parkinson's disease, with model membranes that try to simulate lipid raft compositions. And the alpha-synuclein is a protein that is unstructured, very abundant in neurons, and it is not so clear what is its physiological function, but it has been proposed a role in the homeostasis of vesicle assembly by binding lipid rafts on membrane of neurons. But it is also involved in the Parkinson's disease, and one of the main molecular events is the aggregation of alpha-synucleins, which leads to the formation of protein inclusion and several cell damages of neurons, leading to neurotoxicity. And regarding the aggregation of alpha-synuclein, we have different factors that can promote the aggregation of the proteins. So in our laboratory, we wanted to focus on a pathological condition, try to correlate the aggregation of alpha-synuclein with a pathological condition, which is the accumulation of iron. It is reported that Parkinson's disease patients are characterized by high levels of iron and other kind of metals. So our idea was try to correlate the ability of alpha-synuclein to interact with, the effect of iron in the ability of alpha-synuclein to interact with the model membranes that mimics this kind of structure, the lipid rafts on our membranes. And we also correlate the activity of the wild-type alpha-synuclein with mutated species which is the A53TT, that is a mutated form of alpha-synuclein carrying a missense point mutation, and we have a amino acidic substitution in the position 53. And it is a protein that usually show a better propensity to deaggregation. So we started with an in vitro aggregation assay in order to investigate what iron can lead to the structure of the protein. And we found an early stage oligomerization of both proteins with a better propensity of the mutated species, both in terms of particles that we found and also for the dimension of these particles. After that, we moved to an investigation of the interaction with our model membranes, and so we compare the behavior capacity of the protein to bind membranes before and after the metal treatment. We produce artificial model membranes that mimic lipid raft compositions. In this case, we used 3 lipids, the phosphatidylcholine, sphingomyelin, and cholesterol. Sphingomyelin and cholesterol are 2 molecules that are enriched in lipid rafts. We used the vesicle fusion method to produce a planar lipid bilayer deposited on mica, and we analyze with atomic force microscopy in liquid the morphology of this membrane. In this case, we have a coexistence of 2 phases. We have a fluid phase, the liquid disorder fluid phase, which is the DOPC-enriched phase. And we have also the presence of domains, which represent the raft domains of our membrane, which protrudes from the fluid matrix of the surface of around 1 nanometer. So after this characterization with atomic force microscopy, we moved to analyze the binding, the membrane binding of the 2 proteins. The monomeric proteins leads to similar defects. In fact, the morphological and the dimension of the defects in deep in the plane of the bilayer, which is in the range of 0.51 nanometers, is compatible to an interaction of the proteins leading to stretching and thinning of the membrane, which is one of the model proposed for the formation of vesicles in neurons. But what is different is that the wild type is able to interact with both lipid phases, the solid order phase and the fluid phase of the membrane, whereas the mutated form just interacts with the fluid phase. And what we observe is a fast morphological reorganization of these solid domains. After that, we decided to see what happens when we incubate our iron-induced oligomers with our membranes. And in this case, we have a completely different effect of both proteins on the membranes. We observe the presence of clusters of oligomers that seems to interact preferentially with the Raft domains, but not on all domains of membranes. So probably there is a specific nucleation of the proteins on the domains with the assembly of this kind of structures. And what we observe is that we have aggressive behavior of the mutated form with the presence of these bigger clusters with this ring shape. And after that, since the aggregation of alpha-synuclein usually is due to not the aggregation of one single type of molecules, we decide to perform co-incubation and co-aggregation assay in order to see if this kind of effect is specific for the mutated proteins. And in the first case, we did a co-incubation of the oligomers of both proteins that were separately formed in liquid. And what we observe is a bigger number of clusters, but appear to have a smaller dimension, as in presence of a very poor interaction between the 2 types of oligomers. Whereas when we did the co-aggregation, so we obtain a mixed oligomers, mixing in same amount the 2 proteins, so we obtain the same kind of structure. So in this case, these mixed oligomers maintain the ability of the most aggressive proteins in the interaction with membranes. So in conclusion, this first part, what we demonstrate is that both proteins, as the monomeric proteins, has different membrane ability to bind these lipid rafts and model membranes. And so the question is if this different ability to bind the raft domains could be in somehow correlated with a different assembly of vesicles. And we proved that the iron is able to induce stronger aggregation. We confirmed that the aggregation is most pronounced for the mutated form of the alpha synuclein, and that the accumulation, the formation of this specific structure by this kind of oligomers could have in somehow be involved in the impairment of molecular pathway of the proteins. I have time, I think. Yeah. Yes. And now I want just to show you some preliminary results of this new project of my laboratory, which is focused on the biophysical characterization of extracellular vesicles and the interaction of these vesicles with model membranes. Extracellular vesicles are cell-derived vesicles that are characterized by a specific molecular content that are used by the cells to communicate, and so they can be considered the perfect candidates for therapeutic treatment. So among the different questions that we would like to resolve in our lab, we focus on the mechanism of internalization of this kind of vesicles. So the internalizations happens at the level of plasma membranes, and it seems that we have different kind of pathways that are involved in this kind of interaction. So we perform a functional characterization of this interaction with our model membranes. At the beginning, we just characterize extracellular vesicles with atomic force microscopy, showing that the liquid characterizations allows to maintain the structure and the morphology of this kind of vesicles, and also it allows to have a morphological and dimensional characterization. This is the preliminary results of interaction of this structure with model membranes made by B-component DOPC and sphingomyelin. So here we have solid order domain and the liquid order phase of the membrane, and after the addition of extracellular vesicles, we observe these kind of structures. So we noted that we have an accumulation of these vesicles just in some part of the membrane. And if we look at higher contrast image, we see that we have just 1, 2, 3, 4, 5 solid order domains that remain free by this kind of interaction. So we suppose that probably this kind of interaction is crucial or is very important for the interaction of the vesicles with membrane. And also another thing is that looking better at the image, so what we noted is that we have these entire lipid vesicles that accumulate, but they are surrounded by this structure that have an age about 6 nanometer and is compatible with a rupture of extracellular vesicle on our systems. So I finished, and I want to thank all the components of my laboratory, our collaboration for the extracellular vesicle project, the SciTrek Lab and Uni Udine, and thank you for your attention.
Thank you for the nice talk, and we have time for questions, so please.
Okay, great talk. Again, I'm not a biologist, but what interests me most is, again, the AFM technique and how you employ it to these biological systems. So you detailed a bit about what you're doing, but what I'm interested in is, like, what do you expect from the AFM technique in order to improve these measurements? I mean, measurements in liquid, or analyzing more mechanical properties for these case of studies. What is your wish list, let's say, for the AFM inventions?
So we would like, the fact is that we have, with our atomic force microscopy, we have a long time in the acquisition of the images. So It's like minutes, 20, 30 minutes to have one images. And especially for the interaction of oligomers, we would like to be high speed in order to see the first temporal, I can say, the first moment of the interaction of this kind of molecule, to be sure that these kind of structures goes to rough domains, and also to see the formation of these kind of clusters. This is the first, if we are to talk about atomic force microscopy, that we would like to do. And for sure, maybe also for spectroscopy, but we are more interested in the dynamics of the processes that take place on membranes.
Yes.
Thank you for your really interesting talk. I was curious that in your experiment you are using the man-made membrane.
Yeah.
So there are no proteins on that side.
No,
So
it's how its membrane is formed. No, is I just show you. No, sorry, I skipped.
I can understand that.
Yeah.
But
No, it's an artificial lipid bilayer. You have just
yeah.
lipids, so you have the DOPC and sphingomyelin and the cholesterol. So you create vesicles without proteins, and then you spread your vesicles on mica. Usually we work with mica. In liquid, you obtain the planar lipid bilayer.
Yeah, I understand that. But my question is, do you think what is the difference between the protein interaction with the pure membrane and the membrane with
for sure, here you have just, I think, electrostatic interaction with your bilayer, and for sure there is a different behavior, I think, in the case of real membrane with proteins. In fact, we are working on a protocol to obtain a fragment of membranes in order to have a planar membrane of a real cell membrane, in order to test this kind of structure, to see what is the effect. Thank you.
Any other question? I would have one. Yeah. Regarding the interaction between the membranes and the oligomers, why you didn't measure in liquid, or simply you are not showing? This was in air, right?
The membrane with the oligomer? No, in liquid. It's in liquid.
It was in liquid. Okay, sorry, it was not clear.
Just the measurement of oligomers alone at the beginning, it was performed in air. The other measurement of membrane and alone and with oligomers and monomers are performed in liquid.
Okay, thank you.
Okay.
Thank you very much.
Thank you.
So I think we can close this section.